Hello everyone, welcome to BioPractify. I am Khushi and today we are diving into a fundamental topic in bioinformatics, that is sequence alignment and analysis. If you are studying biology, genetics or working with DNA or protein sequences, this is the essential knowledge. So let's get started. What is sequence alignment? Sequence alignment is the process of arranging DNA, RNA or protein sequences to identify regions of similarities. These similarities can tell us a lot about evolution, conserve biological functions and even structural prediction. It helps in revealing evolutionary connections. By aligning sequences from different species, we can trace shared ancestry and evolutionary divergence. Second, it helps in identifying conserved motifs. Conserved regions often indicate functional importance like enzyme active sites. Third, it helps in predicting structures and functions. Similar sequences often mean similar 3D structures, which helps in predicting protein function. And last, supporting comparative genomics. Large-scale comparisons across genomes helps us ennote genes and understand their organizations. Every DNA and protein sequences holds the blueprint of life. By understanding these sequences, we unlock the mystery of living organisms. It helps in decoding genetic information. DNA contains instructions, protein execute them, and alignment helps interpret these instructions. Second, evolutionary insights. Comparing sequences helps reconstruct phylogenetic trees and understand adaptation. Third, impact of mutations. Alignment lets us locate and analyze mutations, key for diagnosing diseases and developing drugs. So now let's see types of sequence alignment. There are mainly two types of sequence alignment, pairwise alignment and multiple sequence alignment. In pairwise alignment, it is further divided into global alignment and local alignment. So in global alignment, it compares entire sequences end to end for best closely related sequences of similar length. The main algorithm that is used in it is Needleman-Wunsch. Second, local alignment. It focuses on regions of high similarity within larger sequences. It is useful for finding conserved domains. And the main algorithm that is used, Smith-Waterman or BLAST. And pairwise alignment. This is the process of comparing two sequences such as DNA, RNA or proteins to find the regions of similarity. It can be applied globally and locally. Last, multiple sequence alignment. It aligns three or more sequences to highlight conserved regions and evolutionary relationships. And the main algorithms that are used are Clustal W and Omega. Before we go further, understanding these terms are important. Homology, shared evolutionary ancestry. Identity, exact matches in residues or bases. And the similarities means bases or residues with similar properties. And the gaps in sequence alignment means insertions or deletions in one sequences compared to another, penalized to avoid excess gaps. And the scoring matrices are PAM and BLOSSOM, are used for proteins, while simpler schemes are used for nucleotides. Now let's talk algorithms. Needleman-Wunsch guarantees optimal global alignment, accurate but computationally intensive. Second, Smith-Waterman finds optimal local alignment, great for conserved domains. BLAST, it stands for Basic Local Algorithm Search Tool, a heuristic algorithm for rapid database searching, trading some sensitive for speed. Clustal W/Omega, it is used for multiple sequence alignment, key for discovering conserved regions across many sequences. Now let's see how BLAST works. BLAST is one of the most popular and widely used tools in bioinformatics. It helps scientists quickly find regions of similarities between a query sequence, such as a gene or protein you are studying, and a huge database of sequences stored in online libraries. The first step is query input. First, you upload your DNA, RNA or protein sequence to the BLAST website. This is your question, the sequences you want to compare against all the others. Second, database selection. Next, you pick which database BLAST should search in. For example, you might choose NR, non-redundant, which has many sequences from all over the world, or SwissProt, which contains highly reliable, well-annotated protein sequences. The result interpretations are as given: E-value, expectations value. This tells you how many matches you could expect to find by chance when searching the database. A very low E-value like 1E minus 15 means the match is highly unlikely to be random, so it's a strong and meaningful match. A higher E-value suggests the match might just be a coincidence. The second is percentage identity. This shows the percentage of nucleotides or amino acids in your question that exactly matches the sequence found in the database, revealing how similar the two sequences are. Third, score. This is a number that measures how good the alignment is based on how many matches and mismatches there are, plus any gaps introduced. For example, if you upload a human gene sequence named gene X and get a hit with an e-value of 1e minus 50 and 99% identity, this means BLAST found a very close relative of that gene, either that exact same gene in another organism or a gene that has been well conserved over time. So now visualizing alignments helps you understand sequence relations. The first is pairwise DNA alignment, shows matches, mismatches and gaps between two sequences. Second is multiple sequence alignment. It highlights conserved regions across a group. Tools like Clustal W and Omega automate this process. Now let's jump to the application of sequence alignment. Where is sequence alignment applied? First, gene/protein function prediction. Identifying active sites and regulatory elements is important in bioinformatics. Hence, it helps in that. Second, comparative genomics and phylogenetics. It helps in studying evolutionary relationships and trace pathogen evolution, such as COVID-19 variants. Third, mutation analysis. It helps in finding disease-causing mutations critical for personalized medicine. So, to wrap up, sequence alignment is a core tool in bioinformatics. It's the first step in many analyses, from gene discovery to evolutionary studies. Algorithms like Needleman-Wunsch, Smith-Waterman and BLAST are powerful and robust. Alignments reveal gene function, evolutionary relationships and disease mechanisms. So before we finish, let's answer two important questions about sequence alignment. How does an e-value tells us if a match is real or just by chance? Okay. So think of an e-value like a score that tells us how surprised we should be to see a match when comparing sequences. A small value means it is very unlikely the match happened by luck. It's real and important match. A big e-value means the match could easily happen by chance, so it's probably not meaningful. In simple terms, low e-value, it is special, and high e-value, it could be random. The second question was the difference between local and global alignment. Why choose one over the other? And I would hope you guys would answer this question in the comments. So if you like this video, please give a thumbs up and don't forget to subscribe for more in Bioinformatics Inside. Thank you for watching and I hope to see your answers or thoughts in the comments. Thank you so much.